Well, first of all, I want to thank the organizers for inviting me to give this talk. So I was asked to talk about diet on the microbiome, that impact, but what I want to do is I really want to extend this to why we should think about diet's impact on the microbiome, thinking about human health and disease. So I'm going to give you sort of a 30,000-foot overview using a number of different types of examples in my talk in the literature, just a little bit about what we're doing at Penn. So this is my 30,000-foot view of the intestinal environment. So topologically, the microbiota exists in the gut lumen. It's the external environment. And the internal environment of the host contains the mucosal immune system in the bottom. And what separates us from the external environment is remarkably only one cell layer thick, and that's the intestinal epithelium. Now, we produce substances, for example, through the intestinal epithelium, mucus and antimicrobial peptides that help to shape the composition and function of the microbiota. In return, the microbiota interacts with us. Certain types of microbes and their products elicit an anti-inflammatory response by augmenting T regulatory cells. Other types of microbes elicit more of a pro-inflammatory response. And bacteria in general help to program plasma cells to secrete specific immunoglobulin A that's secreted into the mucous environment to fortify barrier function. For the purposes of my talk, it's important to know that what you consume, what you eat in terms of diets, antibiotics, and xenobiotics not only help to shape the composition of the microbiota, but diet in particular can be utilized by microbes to produce a whole panoply of metabolites that we cannot normally produce, that we end up actually absorbing, that circulates systemically. And so the notion is that together with the ability of the microbiota to shape mucosal immunity, along with the fact that it's producing these metabolites that we absorb, may be playing a role in increasing incidence of many different types of diseases that we see associated with industrialization, and this is only a partial list. Now, there's a profound increase in these types of disease processes that we actually see globally. These are not due to our genetics, because we don't evolve that quickly. This is clearly a very strong environmental effect, and the notion is that because a lot of these disease processes in some way are associated with inflammation, it's given the field the notion that perhaps there's something different about our microbial environment. That's a notion of the hygiene hypothesis, which is what I show you on this slide here. So if you're an infant born into a normal microbial environment, you establish the normal microbiome. And because of all the reasons that I showed you on the previous slide, you develop a normal mucosal immune system, which means that you have a normal immune tolerance. In other words, you get infected with an organism, you have inflammation, you clear that organism, inflammation goes away and you're largely healthy. But if you're an infant born into an abnormal microbial world, and our microbial world is fundamentally different now than it was decades or generations ago. We're living in a much more sanitized environment, we're using antibiotics, diets are different. There's many different reasons why our microbial environment is different. We actually know this because when we sort of look at the microbial environment of people living in agrarian cultures, in isolated tribes in Peru, for example, versus industrialized nations, they are fundamentally different. Because of this, we failed to establish a normal microbiome, and then we developed this inflammation, autoimmunity, prone immune system that somehow later on in life, due to another environmental cofactor, we now begin to respond abnormally to microbial products and their autoantigens, and we develop these chronic inflammatory processes. So if this notion is correct, actually early life exposures to microbes may be fundamentally important in predisposing for the development of immune-mediated diseases later on in life. We actually observe these associations based on clinical epidemiology of human populations, for example, being born by cesarean section, or the early use of antibiotics, or formula-fed infants seem to be at higher risk for diseases such as atopic disease. And there are many different animal models that suggest that early life exposure to microbes and with that altered microbiota may predispose to the development of disease. So unfortunately, I don't really have a good pointer here, but this is work by Marty Blazer, just as one of these several mouse examples. So what Marty had actually shown is that the treatment of mice with antibiotics leads to obesity in mice. And we know that it's because of alteration in the microbiota, because you can transfer the microbiota of that obese mouse into a germ-free mouse, and that germ-free mouse now becomes obese. Relative to a mouse that actually never saw antibiotics that's relatively lean, when that microbiota is transferred into a germ-free mouse, the germ-free mouse remains lean. The remarkable thing is that if you treat a mouse early on in life with a single pulse of low-dose antibiotics, that mouse is now susceptible to the development of diet-induced obesity later on in life, suggesting that, again, early on in life, that alteration in the microbiota may predispose to the development of disease. Now, we know that the microbiota early on in life is actually very dynamic for the first 2 years of life until it actually matures at about the age of 2 or 3. So you can see that there are many different types of colors here. It's very, very dynamic. And one of the biggest changes actually occurs with the introduction of table food. So this is the composition of the microbiota in a single infant over a 2-year period of time. So that was done a couple of years ago, but I show you this study by Frederick Backhead, where he did shotgun metagenomics of the microbiota in 98 mothers and their infants. And he showed a couple of things. First of all, he confirmed that delivery mode has an impact on the microbiota. Born by C-section or vaginal birth has an impact on the microbiota. He also shows that as the infant grows older, there's an increase in complexity of the microbiota. That has previously been known. But what's actually quite interesting was that it was the cessation of breastfeeding rather than the introduction of solid foods that led to the maturation of that microbiota. So I showed you on a previous slide, our notion had previously been it's the introduction of solid foods that led to the maturation of microbiota. What his data actually shows is actually the cessation of breastfeeding, and it suggests that breastfeeding, breast milk, helps to maintain the microbiota in an immature state. Part of that may be through human milk oligosaccharides that are in breast milk that are deeply bifidogenic, that promote the expansion of bifidobacteria, and when you take that away, other types of microbes may be able to invade, and you see this maturation of the microbiota. So it's a very interesting observation that might have an impact, given the difference in various types of diseases associated with modalities of early infant feeding. And when you think about it, breast milk is the most fundamental, well-defined nutrient food that we consume. So extending that a little bit further is work by Jeff Gordon. So Jeff Gordon has led to seminal advances in the field, and Jeff's lab for many years has actually been quite interested in the microbiota as being a source to perpetuate malnutrition on infants in the third world, for example, in Malawi. In this study, he actually tried to understand exactly what it is about that microbiota that's different. And what he actually found was was the milk. So using metabolomics screen of mother's milk, of mothers who had malnourished infants versus well-nourished infants, he found that the mother's milk in malnourished infants was different in that it had a decreased content of these fucosylated milk oligosaccharides, what I mentioned just previously on a previous slide. And to show the functionality of this, he actually took the microbiota of these underweight Malawian infants and put them into germ-free mice and germ-free pigs. We established that microbial community and then fed these animals the prototypical Malawian diet supplemented with these sialid milk oligosaccharides, and could show that in fact these mice and these pigs would actually grow better with greater lean mass, better bone growth, metabolic function, suggesting that in fact it may be the diet, primarily mother's milk, working through the abnormal composition of the malnourished Malawians' infants' microbiota that is playing a role in underweight infants in this part of the world. So our microbiota interaction with diet is actually quite complex. So I show you on the left here one of our first studies that we published a couple of years ago. And this is a heat map essentially making an association between different types of micronutrients along the y-axis with different types of bacteria along the x-axis. So the positive correlations are in red and the negative correlations are in blue. And the intensity of the color indicates the strength of the correlation. Now there are different patterns of color here. I'm not going to get into depth, but I want to call out two genera. One is Bacteroides, and the other is Prevotella at the top, and you can see the arrows, and you can see visually that the colors are basically opposite from each other. So the associations with various micronutrients associated with the abundance of Bacteroides versus Prevotella, one is blue and the other one is red. Okay, so just keep that in the back of your mind. I'm going to come back to that in my next slide. Now, there are a number of studies that show that there is an association between the microbiome, diet, and disease. And that is, there's a decrease in microbiome richness. This is what Vince had mentioned. That's a decrease in the number of various bacteria and their genes that are associated with both disease states and a consumption of a westernized diet. So what do I mean by that? Individuals with marked obesity, insulin resistance, dyslipidemia have low bacterial richness. Individuals consuming an agrarian diet have increased bacterial gene richness, and energy-restricted diets increase bacterial gene richness. And this is actually what we see globally, where individuals living in Africa have a richer microbiota than individuals living in industrialized nations, and they have a decreased incidence of many of these metabolic and inflammatory diseases that we see associated with industrialization. So one of the holy grails in the field is to try to categorize people by looking at microbiota composition. And this is a study. It's controversial, that was published by the European MetaHit group. So the European MetaHit group is a group that's sort of like the counterpart of the Human Microbiome Project group in the United States. So they had coined this term enterotypes in the gut microbiota. And in other words, you could classify people by dominant taxa, one being Prevotella, one being Bacteroides, one being Ruminococcus. And you know, the value of this is very significant, because if you could classify individuals, you could maybe make some type of prognostication about the development of disease, or you could think about response to diet, et cetera, et cetera. So it's controversial in the sense that the computational biologists have been fighting for years as to whether or not these are actually discrete, or it's more of a continuum where people with high levels of Bacteroides or Prevotella are sort of at the extremes. I tend to feel that it's probably sort of more of a gradient, but it actually misses a fundamental point that, that we actually see globally. And this has actually been reproduced in many different studies. So individuals living in agrarian cultures, for example, in Africa, Malawi, Bangladesh, have high levels of Prevotella, whereas individuals living in the United States or European Union, more industrialized nations, don't have very much Prevotella. They have higher levels of Bacteroides. So this is actually similar to what we see globally. We don't understand the biological reason for why there's this difference in Bacteroides and Prevotella. I can tell you, we study individuals, vegans and omnivores, and it's actually very rare for us to find an individual with predominant Prevotella. So they seem to co-exclude each other in the environment of the gut. The reason for this remains unknown. But why would it be of value to know the abundance of Prevotella or Bacteroides in the human gut? Well, this is another study by Frederick Backhead's group. And it's bringing home a point about metabolic function. So it's actually been known for a long time that the consumption of undigestible carbohydrates, and in this case they studied barley kernel carbohydrates, seems to improve metabolic function in terms of glucose tolerance. What he actually showed both in mice and actually in humans was that the abundance of Prevotella in the microbiota was a predictor of how well you would actually respond. So if you have high levels of Prevotella, it's more likely that you're going to respond favorably to food supplementation with these complex carbohydrates in terms of glucose tolerance. So that's actually quite interesting, because it means that, well, perhaps you could predict who would be better SERD for treatment using a dietary intervention. Thinking about it further, well, maybe you could engineer the microbiota to have more prevotella so people would respond more favorably to metabolic syndrome with dietary supplementation. It's just a notion about this, but we're beginning to see this more and more in the literature. So personalizing responses to diet based on the structure of the microbiota. And so people are using the power of computation as well as metabolomics along with describing the composition of the microbiota to come up with models that help us help us determine how you would respond to diet. And so I think you're going to be seeing more and more of this as people develop more computational tools, as the cohorts larger and larger, they're being studied. Okay, so I think this is one of those things that we're probably going to see that's going to be actionable sooner than later. On this slide, I make the point that what you eat in terms of diet not only changes the composition of the microbiota, but also, again, serves as a substrate for the production of all these small molecules that after first pass metabolism circulate widely throughout the body and could have effects at organs distant from that mucosal environment. So an example of this is of metabolite production through the gut microbiota and impact disease has been demonstrated by Stan Hazen. So Stan has shown that the consumption of dietary fat as well as carnitine delivers choline to the gut microbiota, and choline is converted to a gas by the microbes in the gut called trimethylamine. And after oxidation in the liver by flavine monooxidases, there's a small molecule called trimethylamine oxide, which accelerates coronary vascular disease in mice and actually is a biomarker for coronary vascular risk disease in humans. In fact, that's how Stan found TMAO, is he did this wide-ranging metabolomic screen of individuals at risk for coronary vascular disease, and he asked, well, where did TMAO come from? And it turns out that it comes from the microbiota. So if you think about this pathway, how might you think about things that you might be able to do to prevent and or treat disease? Well, microbiologists have identified the gene that converts choline to trimethylamine, that gas, and it's called the CUT-C gene. It's a gene in the family of the choline TMA lyase family. So if you know the gene, that you can go back through databases and identify the microbes that have that gene that could make that conversion. And it turns out that most of the organisms that are commensal with us that can make that conversion live in the gut. So if you know the gene and you know the bacteria, how might you think about innovative strategies to prevent and treat human disease? Well, you quantify the risk for heart disease by characterizing the abundance of bacteria in the gut that have this CUT-C gene. You could develop medical foods to reduce the production of TMA by bacteria from the diet, or you can reduce CUT-C expressing bacteria in the gut, or develop drugs to inhibit CUT-C activity in bacteria. And what I'm going to show you in the next slide is some work by Stan Hazen, just recently published within the last year, making this specific point. So what Stan has shown is that there is a choline mimetic. It's called a dimethylbutanol that doesn't kill bacteria, but what it does is it actually inhibits TMA lyase activity, inhibits the ability of bacteria to convert choline into that gas, trimethylamine. And he shows that in vitro, as well as feeding mice with DMB, reduces TMAO production and reduces coronary vascular disease, at least in mice. So it's a very interesting notion. We think about small molecules, drugs, as things that affect us as the host, but it's a notion that you could develop small molecules that would actually target your microbiota to change its functionality. It's a very interesting concept, but I always pause here because I get these questions where people say, well, so Gary, what type of diet should I eat so my microbiota doesn't cause heart disease? And what I remind people is that we actually know a major factor in the development of heart disease, and that's dietary fat, right? So lipid levels, LDL, HDL, that's why your doctor looks at these lipid profiles, and that's why statins are one of the best-selling drugs in the world, because they actually work to reduce the risk of coronary vascular disease. These types of concepts that I show you are basically made in animal models with associations in human biology. What we don't understand is the relevance of these types of models to human biology. And if it is relevant to human biology, what's the effect size? It could be not relevant to human biology. On the other hand, it could be relevant, but it have a very small effect. On the other hand, it could have a profound effect. We don't actually understand that route. It's very important that people understand that. And I think that's a challenge for scientists and physicians in the field, is really to answer that question over the next decade. Nevertheless, we see these very interesting signals in new biological pathways, and they may be actually very profound. So when we think about the metabolites, obviously most of the metabolites, the input is through diet, directly on us as the host, but definitely diet can have an impact on our metabolome through the gut microbiota. But an important point to make is that what we measure in plasma is actually at steady state. It's not only what's coming into our body, but it's actually what's actually being excreted. And one of the methods by removing these molecules from the body is urinary excretion. So it's actually in an equilibrium. I'll show you very quickly about a study that we have to characterize the human metabolome from the microbiota that we call PharM. And we're embarking in a study to really understand the impact of decreased renal clearance on these types of microbial metabolites in a large cohort of individuals with chronic kidney disease. So we are interested in trying to understand that equilibrium. Why might that be relevant? Well, in chronic kidney disease, as kidney disease progresses, there is a profound impact on coronary vascular disease. It's not actually well understood why this actually occurs. It's a multifactorial process. Nevertheless, individuals with chronic kidney disease are at very high risk for coronary vascular disease. And the interesting thing is that TMAO, small molecule that I was talking about in terms of Stan Hazen work, is actually cleared by renal clearance. And so individuals that have end-stage kidney disease and hemodialysis have actually very high levels of TMAO circulating in the blood. So it's just one example by which potentially small molecules that may be a disadvantage to the host may have a higher abundance due to decreased clearance from the body. So in my last couple of minutes, let me just focus on inflammatory bowel disease to make a particular point here. So I'm a gastroenterologist. Inflammatory bowel disease, it's a chronic inflammatory disease process, leads to extraintestinal inflammatory disease. This affects a lot of people in the world, and is treated by basically lifelong immunosuppression. We know that the microbiota is dysbiotic. It's abnormal in community structure compared to health. And I don't have time to get into this, but based on animal models as well as some data in humans, that dysbiotic microbiota associated with disease may be helping to perpetuate the chronicity of inflammatory bowel disease. So our strategy from thinking about engineering the environment of the gut for treatment of disease is to somehow alter that dysbiosis, and in many different ways that we can think about doing this. I'm going to focus for the last couple of minutes on dietary intervention. There actually is a dietary intervention for the treatment of Crohn's disease, and that's the use of an elemental diet or defined formula diet. This is used as first-line therapy in Europe and in some places in Asia, a couple places in Canada and the United States. Unfortunately, most of these diets are not palatable, so they have to be delivered by nasogastric tube, and then you pull the tube out during the day, and you're not allowed to eat any solid food. You can only take this as a sole source of your nutrition, and it works very well. But if you start to eat whole foods, the effect goes away. Despite its effectiveness, we really don't have an understanding of how it works. So the idea is here that there are epidemiologic associations between diet and IBD, and for reasons that I don't have time to talk to you about, we know that the gut microbiota is fundamentally important in pathogenesis of IBD. And I just showed you a couple of lines of evidence that diet has an impact on the gut microbiota. So a lot of us are very interested in understanding the principles by which diets that we already know, although difficult to use, how they actually work in treating IBD, because if we could understand their best principles, we would be able to design better diets for patients with inflammatory bowel disease. So we at Penn and Children's Hospital are interested in studying this. I'm just going to show you two snapshots of this. So we have recently published a very large cohort of pediatric Crohn's disease patients that were either treated with anti-TNF therapy or diet therapy. It's a longitudinal prospective cohort. We sampled multiple times and we shotgunned metagenomics to really understand the impact of diet on disease. just as a snapshot of this, what we actually found was that dysbiosis, the microbiota associated with disease, is actually much more complex than we thought. So there are independent factors that independently have a separate effect on that dysbiosis. Disease has an effect, antibiotics has an effect, diet has an effect, and inflammation has an effect. And we would not have uncovered this had we not done a prospective cohort analysis. But the point here is in terms of diet, that elemental diet definitely has a significant impact on the composition of the microbiota. I don't have time to get into it, but interestingly, it was the thing that actually reduced fungi in the gut, and there is a notion that fungi in the gut may be important for inflammatory bowel disease. So what we're finishing up now is another study, and again, because we're talking about diet, I just show you this particular slide. It's called FARM. It's a nice demonstration of how the food industry, principally here Nestle, and two private foundations, Helmsley Charitable Trust and the Crohn's Colitis Foundation of America, have worked together in a partnership with academia to really understand the impact of diet on health and disease. So in this study, the objective was to determine the relationship between dietary composition, gut microbiome composition, and metabolic products that ultimately are present in the lumen and the plasma of humans. So it's a very intensive 2-week inpatient stay where we lock people up for 2 weeks, and it's a controlled feeding experiment. We have vegans, we have omnivores consuming that exclusive enteral nutrition diet. We're interested in understanding the impact of those diets on the microbiome, but just as importantly, the impact on the universe of small molecules in human plasma that is through diet and the microbiome. Now, that's in healthy human subjects. We've done something similar to this in patients with Crohn's disease, and we think that the intersection of these two very large data sets will help us understand in what way diet alters the composition of the gut microbiome, its metabolome, and the host metabolome in a way that reduces disease in patients with inflammatory bowel disease. This study is part of the Crohn's and Colitis Foundation's Microbiome Initiative. The initiative is to identify new therapeutic targets, and our contribution to this consortium is to use diet as that therapeutic probe to identify new therapeutic targets. So in summary, diet has an effect on the host. Diet has an effect on the microbiota that could impact upon the host. When we think about diet's impact on the microbiota, it has a lot to do with the composition of the diet, whether or not it's short-term, long-term diet. It has an impact on bacteria that can be described in many different ways. But there's a notion that it could actually affect the other types of microbes that live in the gut. And we don't really have time to talk about this, but there are these associations that are actually quite interesting. Impact of diet on the microbiota is not simply on the composition of the microbiota, but it's also on the small molecules that they produce, that both of which may have an impact on the host. So when we think forward in the future, we could be developing medical foods that directly affect the host. We can think about the next generation of prebiotics that not only shape the composition of the microbiota, but serves as that substrate for metabolite production by the microbiota. You could think about the next generation of probiotics that more deeply change the composition of the microbiota for treatment or prevention of disease, but maybe it's not good enough to simply give the substrate, but you have to have the right microbes in the environment to convert those substrates into those small molecules, and that's that notion of symbiotics. And just as an acknowledgement, we have an entire team that has to work together. It's really a team research, so Rick Bushman and Jim Lewis and I co-head the microbiome team, the intestinal microbiome team at University of Pennsylvania, and I want to especially acknowledge Bob Baldessano at Children's Hospital that really worked with us on the elemental diet study. So thank you very much.
Thank you for the wonderful talk. I just have a quick comment/question. You already mentioned that enterotypes are tricky, and so I'm not going to touch on that. But you said that you don't find a lot of Prevotella in US composition. So I'm wondering what kind of population did you study? And the reason I ask this is because we have done a study on healthy or otherwise healthy control population versus people who suffered enteric diseases, and we saw that the healthy population had Bacteroides, as you mentioned, but the sick population had more Prevotella as compared to them. So I was wondering what your study population was.
Yeah, so I was commenting on healthy human subjects. And so we have fairly large cohorts, 100 patients. We've worked with Stan Hazen on his cohorts also. And we've actually even looked at omnivores and vegans, very distinct diets. And we find Prevotella predominant individuals very, very rarely. We would love to find more, but we can't find them.
Quick question. Have you or anybody else done a study, provided it's within the animal rights or animal control, ethical use of animals, to actually deplete the diet intake of mice and see whether when you disrupt the diet and then restart the diet, does the microbial diversity, richness change when the microbiome comes back, or does it get completely changed? Like if somebody goes, you know the question what I'm asking.
You're talking about mouse studies then?
Yeah. Human studies, obviously, I don't think any IRB would allow you to do that to almost to the level of starvation and then bring it back up.
Right. So Jeff Gordon has actually shown this. I have 2 comments on that. So Jeff Gordon has actually shown that dietary restriction will shift the composition of the microbiota in terms of caloric restriction within 12 hours. Okay.
No, I'm not talking restriction. I'm talking of starvation.
Starvation? We haven't done those studies. I'm not aware that people have done that. The only thing I can say is that there are individuals that have been studying total parenteral nutrition, who are essentially not eating anything, basically being fed to the vein. And there you can also see a significant composition change in the microbiota. But I do have one caveat to say, that in mice, and I'm a mouse researcher, so I love mouse research, okay? In mice, change in diet has a massive effect, almost at a phylum level, on the composition of the microbiota. It's actually very consistent within mice. Okay? In humans, we've done a study, a healthy human subject study, a feeding study, which showed that humans don't respond in the same way to diet as mice do. So the effect, it is really there. So what you eat will change your microbiota within 24 hours, but the effect is relatively modest, and it's not predictable. People move in different directions, almost as if where you move to, so it depends on where you start. So we have to be very cautious about extrapolating too much from our mouse studies in humans. I'm a big fan of doing intervention human studies in parallel with mouse studies to look at relevance between the 2 model systems.
Thank you.